We know now that many psychiatric disorders are a consequence of abnormal brain development. This is very obvious for disorders that start early in life, like in the case of autism. And it's less so obvious for other disorders like schizophrenia, for which we have to wait almost 2 decades to have a clinical entity. However, there is something common among all these disorders, and this is the realization that they are a consequence of brain development going wrong in one way or another. The new MRC Centre for Neurodevelopmental Disorders, we have the opportunity to bring together clinical experts with basic neuroscientists to basically understand these mechanisms, what could be causing these disorders, and how these particular gene variations and environmental insults could eventually modify brain development to give rise to these particular conditions.
This new MRC Centre at King's pulls together elements of expertise at Guy's, St Thomas's and Denmark Hill. So you can imagine the team at Guy's will be looking at the petri dish at how neurons are built. Then that technology can be taken forward into how babies' brains are built at St Thomas's. And then down at the Denmark Hill campus, we're looking at how this manifests as a condition such as epilepsy or schizophrenia or autism in later life.
Genome editing is a very new technology to change very specific sequences in the genetic makeup of any organism, including human cells.
Together with our clinician colleagues, we have started to generate animal models in which we can test these specific mutations that we have found in patients and trying to understand better some of these very devastating disorders.
And the combination of human studies that we can provide and the very deep scientific experiments that they can carry out is by far the most powerful way to try and understand what's going wrong with brains of children who don't develop normally.
What the Centre will allow us to do over the next 5 years is really address crucial questions. For example, why is it that the same genetic abnormality in one human leads to so many different outcomes, potentially to psychosis in one individual, yet epilepsy in another, or autism in another? So what is it that determines that risk versus resilience for developing disorder?
We also aim to inspire and nurture the next generation of world research leaders capable of working in the interface of fundamental science and clinical research.
So we have a fantastic new PhD programme. PhD students will spend the first year doing 3 rotations in labs of their choice, which is a really fantastic opportunity to try out different techniques, experience different kinds of research with really outstanding scientists and clinicians in this field. And at the end of this period, students will get to choose where they want to do their PhD, which I think gives maximum choice to students and really allows them to experience the whole gamut of different types of research techniques available in the centre.
It's also very important to understand how these disorders progress during development from early on to the time in which they manifest all their clinical problems, because we think that early intervention or perhaps early treatments may actually help to improve the quality of life of these patients.
If we can come up in the next 5 years with a novel way of diagnosing autism, schizophrenia or subtypes of epilepsy, then I think we'll have achieved a massive success. At the moment, so many autistic subjects are diagnosed too late. So many epilepsy patients, one might envisage, not treated with the appropriate treatment early on in the disease course. And in schizophrenia, we have all these people at risk of developing psychosis, and then we may not know until the late teens and early 20s who might develop it. So I think the imaging is going to really help us to get a handle on one or more of those conditions.
The imaging facility that we have enables us to take very detailed pictures of how the brain is structured and also how it functions in babies as small as 23 or 24 weeks, and indeed in babies before they're born. And this ability to map the brain at this very early age offers huge potential in understanding how it develops normally, and by understanding that, how it develops abnormally and leads to neurocognitive problems.
King's College London is a unique place to carry out translational research. We are the most impactful of any mental health research group in Europe, and we're the most impactful of any research group in autism in the world. And what we're able to do is to translate our findings from the clinic into basic rodent and cellular models, and then forward translate those findings from rodent models and cellular models back into the clinic. So we can ask those really essential, really crucial step change questions. What is it that causes disorders, and what can we do to reverse them?